Let me record this so I didn't record them. So, today's organismal development part 1. Tomorrow will be 2 and so forth. And then, let's see, next Thursday should be your test on endocrine system and organismal development. Okay? I would expect a quiz on Monday on the endocrine system where you have to match descriptions of either endocrine organs or hormones and what they go with. Also, I would think on that quiz you're going to have to label where all the different endocrine organs are. I think all of them are on that page 999, except for, did we talk about the thymus? Maybe we didn't. Oh, we had a couple minutes left in that Bozeman video the other day, didn't we? Yeah, so all of them are on 999 except the thymus. The thymus is a gland that is found, it's under your sternum, behind your sternum, and in front of your heart. So if you think of your heart being like right here in the middle of your chest, your thymus is right in front of that. Thymus is primarily involved in development of T cells, hence why they're called T cells. And like helper T cells, cytotoxic T cells. And the weird thing, they say after puberty, the thymus kind of like degenerates and becomes kind of fatty tissue after that, or like it's not really functional after that point. I guess you've made all the ones you needed to make, and there you have it. But anyway, if that finds its way on there, you'll know where your thymus is. All right, so test next Thursday. Hopefully y'all will enjoy this unit. I know y'all started watching the Miracle of Life yesterday, and I think all of you were born through a similar process, and you'll probably have kids one day, and it's important to know where babies come from and how they get here, and it is magical and a little disgusting at the same time. So if you have any questions, that's what I'm here for. So the last place you want to be learning about where babies come from and how babies develop and your menstrual cycle and everything else is when you go to the doctor's office or looking on the internet. So let's make sure we get all that sorted out now, Walker. All right, important concepts from previous units. Number 1, Mariah.
Cells that divide by mitosis produce genetically identical offspring.
All right, so we can say they're basically what? What do we call those 2 identical offspring from mitosis? Daughter cells. So they're essentially clones of the original cell. Genetically should be identical unless there's some weird mutation. You're hanging out in the sun too much, or I don't know. Number 2, Matthew.
The cells divide when they become inefficient at transport across the cell membrane.
All right, so as a cell becomes larger, what decreases?
What increases?
What decreases?
Their efficiency.
Functional efficiency. Yeah.
More specific.
The surface area to volume ratio.
As a cell becomes larger, the surface area to volume ratio becomes smaller. And so it becomes less efficient. Remember, the smaller the surface area to volume ratio, the more efficient a cell is at moving waste out and nutrients in. Good. So, good. You getting all this, Matthew? I know some of y'all just have your notes on your phone because of something that happened, but I would prefer if you print off your notes, you people at home too, so that you can annotate and write in, and then if they're easy to refer back to when you're finished. I know we have to kill a few trees in the process, but I planted enough trees in my lifetime to sustain this entire school for paper, so it's okay. Trust me. Number 3, Sarah.
Cells that reproduce sexually depend on cell-to-cell communication/contact.
Now, when you guys watched the sperm and the egg come together yesterday, did y'all watch that? Sperm and egg come together? There's some major cell-to-cell communication going on. Did they talk about what has to happen during that process, or did they just say it enters the egg?
Like, what do you mean?
Like, did they talk about like how you have like 50 or how many ever sperm like around the egg trying to get in, and how the egg only manages to get one in.
Yeah.
What do they say?
So that it created some thing around it to shut off the others from getting.
Yeah, and so when we get to the sperm and egg fertilization part in the notes, and probably like 4 or 5, we'll discuss that a little bit further, because basically what happens is the cell, the whole signal transduction pathway, the sperm kind of acts like a ligand in that scenario where it docks with the egg. And then when it does, it shuts off all those other like dock receptors so that no other sperm can get in. So it's a really efficient way of making sure you don't have multiple sperm entering the egg at the same time.
So do they just like choose the first one, or they just pick?
Yeah, I think it's a first one here kind of scenario. I don't know that you could. A sperm is only made up of DNA and mitochondria and a little bit of sugar and a tail. So I don't know that you could distinguish any admirable features from one to the next if you were the egg. I mean, just think about like what they started out with 200 million of them. If you're one of the 50 or 100 or 1,000 or whatever the small number is that actually makes it to the egg, you're in the winning swimming team. You know what I mean? All right. So number 3, Tay. No, number 4, Tay.
All matter and energy for growth and development comes from the environment.
Okay, so and what that's trying to say, I did a poor job of explaining that this morning. That's really referencing the environment influences sexual reproduction, which is essentially what almost all of the evolution section is on at the end of the year. How does the environment affect your genetics, especially of your offspring? Okay. Modes of doing things. So, sexual and asexual reproduction. Asexual, read that for us, Mace.
Basically, the making of genetically identical clones. No exchange of DNA occurs.
Now, there are organisms like fungi and bacteria and a few others that can undergo sexual and asexual reproduction. So in asexual, you're making genetically identical clones. What are the benefits of asexual reproduction if you're like a bacteria or fungi?
Spread faster.
Faster. So you can occupy an environment much faster. So you would want to asexually reproduce in what type of conditions? Good conditions. If it's good for you, whatever, bacteria, fungi, whatever, then you want to colonize that area and grow as quickly as you can. So the best way to do that is asexual reproduction. You can take over. Now, we've talked quite a bit about how bacteria and fungi are in kind of like a war with each other all the time. Did we discuss that? We talked about like yeast infections and jock itch and athlete's feet and all that. So like even on your bodies, yeast, or well, not just yeast, different bacteria and fungi are trying to occupy your spaces. The same thing's going on in the environment. Bacteria and fungi are trying to occupy the same spaces. And then another war that's taking place is bacteria and viruses, and we'll get into that war in a second. All right, sexual reproduction. Walker, tell us about that one.
The fusion of two different gametes, sperm and egg, to create a zygote, fertilized egg, that it has variation.
Okay, so in the video yesterday, y'all saw the sperm and the egg come together. Now, if you guys, how old are y'all?
17.
If you will add however old you are, and let's say roughly add about 9 months to that, you can imagine back to your first days of life on Earth when the sperm and egg came together from your mother and father and formed you as a young zygote. Just imagine you were once one cell. Okay. And then that cell divided, and it divided some more. And the DNA in that original cell, Mariah, should be the same as the DNA that you have in your body cells today. Unless, you had some weird mutations or been smoking too much or, I don't know, been out in the sun. Some cells obviously there's going to be some mutations that show up for various environmental reasons, but for the most part, should be the same as the ones you started out with many, many years ago. What's up, Matt?
What about twins?
What kind of twins?
Both kinds.
Identical twins. DNA should be exactly the same. So identical twins are, you have a sperm and egg, one sperm, one egg come together, cells start dividing, which we'll see, I think, in section 5. And then for some reason there's some type of spontaneous split that takes place. So the DNA, the cells would all be the same. If you aren't you a twin? Yeah, so you're a fraternal twin. So you and Megs. So you guys, that would have to be, how did that happen?
Wouldn't it be 2 eggs?
So yeah, so you would have 2 sperm and 2 egg, and so 2 different sperm, 2 different eggs. So you guys should be no more genetically related than any other brother or sister. It's just normally what happens is like fraternal twins. So I don't know if y'all caught this in the video when the sperm were trying to head toward the egg. You could be the best swimmer in the world, Walker, and when you get up there, you have to go up. You have to go through the cervix, which is the opening of the uterus. So remember, the uterus is where the baby is, but at the opening there's a muscle at the bottom called the cervix, and so you have to make your way through there, and then you have to go up, and then you have to go down the fallopian tubes, right? So even if you're the best swimmer, you get up in the uterus, Mason, and you get up there and you're just swimming along. Maybe you're first in line. Robert Frost, two roads diverge in a yellow uterus, and you take the one less traveled and you don't get to fertilize the egg, right? So I hope that's okay for the internet. So I mean, you could pick the path down one fallopian tube, and an egg doesn't release that month. Because normally what happens, how many eggs are released? One. And women have how many ovaries usually? 2. So you don't know which one's getting, they're supposed to be tagging each other out, but maybe in the case, now I don't know about your own personal situation, but that would either mean, I would suspect that mean that both ovaries released an egg that month. I don't know how likely it is for one to release 2. It's good extra credit, but I would suspect it would be one of each, and then both of those would get fertilized. And so, like I said, genetically, in terms of relationship, you guys aren't any closer related than any other siblings. Now, a lot of times you see boys that are fraternal twins, they look a lot alike, or even girls too. But I think that's just because genetically you're already pretty close, and you were raised in the exact same environment, basically, and so you wind up looking very similar. All right, sexual reproduction, number 1 and 2. Read that for us, 5. Remember,
variation is the key to survival in changing environments. To create sperm and eggs, certain diploid cells have to undergo meiosis, which involves 2 divisions of chromosomes to become haploid cells.
so we're going to get into meiosis when we get into Mendelian genetics in January, so I'm going to hold off on that. That's basically just how sperm and egg are produced, and it's kind of like mitosis times 2, sort of. But we'll get back. Just know that that's how sperm and egg are made. What was I going to talk about? Variation in changing environments. So if you're like a bacteria or a fungi, and you can have sexual or asexual reproduction, Tay, when would you want to have sexual reproduction?
In a specific time, like not necessarily a specific time, but in a specific environment.
What kind?
Like a warm environment.
Mariah? Matt? Anyone?
Harsh environment.
Harsh environment. Why?
Because there's more genetic variation, the more likely it is like you survive.
Good. So if you are living in a harsh environment, meaning it's hard to survive, and you undergo sexual reproduction, you're creating a variety of offspring. So think of somebody that's got 4 or 5 or 6 kids, and you have all these different looking individuals. Maybe they're different heights and different strengths and stuff. then it's more likely that one or two of those individuals will survive and pass on their traits. Whereas if it's harsh for you and you're having a hard time surviving and you undergo asexual reproduction, your identical offspring are also going to have a hard time surviving. So that's why in harsh environments you undergo sexual reproduction to create that variety, and hopefully some of those individuals who are best fit will survive. That make sense? Super. All right, types of asexual reproduction. Katie,
Binary fission, term for bacterial reproduction.
bi means,
Two.
fission means split. So binary fission is where you have a bacterium split in two. I would think it'd be a good idea for you guys, we don't have time, to make a Venn diagram of binary fission and cell division with mitosis, because there are a lot of similarities here. They're very similar, but obviously bacteria or prokaryotes don't have a what? Nucleus. So that's the big distinction there. But there are a lot of similarities in the way they do it. And we're going to get into the details here in a second. But that's how you make basically little bacteria clones inside your bodies, I'm sure. If they're trying to occupy and take over, they're probably cloning away binary fission-wise to occupy as much of that area as they can. Budding, that sounds fun. Read that for us, Miranda.
Organisms such as polyps
Mm-hmm.
are generating genetically identical buds that break off to become a new organism.
Okay, are y'all familiar with the biscuits you get from the store that have the different layers? You know, they're real flaky and have a bunch of different layers you can peel off? Okay, they're the delicious one. I should bring some of those in. Where was I going with that? Oh, budding. So the polyp is the sessile form, meaning it's attached. So I know y'all have seen jellyfish like floating around the water. Well, they have two life stages. They undergo like a sexual reproduction, and then this zygote then like attaches to the sea floor, and then it starts to grow into this polyp. And this polyp is like the form that's attached. And at the top, like the layers of that biscuit, when one of those polyps develops enough, then it like kind of peels off, and then it floats away into and grows into a normal jellyfish like we see. And there's another one on it, it breaks off and goes away. And all of those that break off like that, just like on a biscuit, they're all identical. They're all the same. So it's just another method of sexual reproduction. Fragmentation and regeneration. Read down for us, Sarah.
Seen in the
Platyhelminthes are the flatworms, which is the simplest phyla of worms that there are. Very, very simple. An easy way to remember it, if you ever see that, is I always think of I always call them platyhelminthes. And I think of flat like a plate. So whatever works for you.
and echinoderms.
Like sea stars or brittle stars or sea urchins or what are those things you collect at the beach? Sand dollars. Yeah, keep going.
For example, a single arm of a starfish gets removed. The starfish will grow, regenerate a new arm in that same place. The removed arm will fragment and become a new starfish.
Yeah, so it's my understanding a sea star or starfish, whatever you want to call it, when part of that limb like gets torn or broken off during like a fight or something, if you've got part of that central portion to go along with it, then like it can grow into a new sea star. And then also that part that broke off will start to regrow. I've got one in the cabinet somewhere. I'll have to look and find it, where you can tell it broke off and then it started to form a new one. I don't know how it would look if it grew a new one from just the foot. I guess one foot would be way bigger maybe than the others early on. But fragmentation or regeneration, we see a lot of fragmentation kind of stuff with similar to what we see in plants, where you can take a plant, a cutting of a plant, and you can grow a new plant out of it. I'm going to try to do that with fig trees for you guys, so y'all have a fig tree to plant at home. Everybody like fig Newtons? Well, good. Then you will like to have your own fig tree to eat and consume. I know I do. Bacteria binary fission. So let's go through the process here. A and B, read those for us, Mariah.
This is the process of reproduction and replication in prokaryotes, bacteria. DNA replication, S phase starts at the origin and works around the entire single circular beta chromosome. This results in 2 identical chromosomes in a nucleoid region.
All right, prokaryotes don't have what?
Nucleus.
So they have a nucleoid region. That's just where the DNA is found. What kind of DNA do bacteria have?
Single chromosome.
Hold on. Yes, they bacteria have one large singular chromosome. They also have other DNA though. They have other little circular pieces of DNA called plasmids. You'll need to know that. All right, we'll get into the significance of those when we get to sexual reproduction. All right, so we have DNA. You have to copy that circular chromosome, and then okay, what happens next? C and D. Read those for us, Matthew.
This is followed by producing a cleavage furrow.
What is cleavage, Matthew? What? Sarah?
It's that little like split.
Okay, it's a split or a break. Good. Keep going.
To produce 2 new cells that are referred to as clones. This cleavage furrow is produced using actin and meiosis microfilaments.
All right, actin and myosin filaments. Where do we see them?
Muscles.
Muscles. So the same cytoskeletal components that make up our muscle and help it contract are also used in helping the contraction and making of the cleavage furrow. So it's the same bacteria using the same stuff we are. All right, keep going.
How is binary fission related to mitosis in terms of evolution? Binary fission would have evolved into meiosis
Mitosis.
as the DNA content increased dramatically, and also have the endosymbiont hypothesis occurred to produce organelles. The 2 major steps are the same: synthesis and division.
All right, so on your Venn diagram between binary fission and mitosis, the 2 things that are similar that are found in the middle would be what? I mean, he just read this. Are y'all high? What's going on here?
Cytokinesis.
Stop doing drugs. Some of y'all can't afford to lose any more brain cells. What?
Cytokinesis.
Synthesis and division. Synthesis of what?
New DNA.
New DNA and division of, Miranda?
The cells.
Cytoplasm. Good. So those 2 things are the same. The major differences is in mitosis, you've got to deal with the nuclear envelope and separation of chromosomes. Okay? But with bacteria, you only have one circular chromosome, and it's just one goes to one cell and one goes to another, so it's pretty straightforward. 100% genetically identical clones are produced by this process. So quick, easy, well, pretty easy. Bacteria, I want to say they can reproduce. I used to do this math thing, where we figured out how many bacteria could grow in like 6 or 7 hours, but we just don't have enough time for that today. But bacteria can reproduce about every 20 minutes. You guys will actually be growing bacteria in the spring in this next thing here. Bacterial variation processes. So we're going to loosely call this like sexual reproduction. I'm going to say loosely call it that because you don't have sperm, you don't have egg. The only kind of way that this relates to sexual reproduction is that there is an exchange of DNA. So, and you are creating more variation. So in that way, it is sexual reproduction, but you don't have the traditional sperm and egg coming together, forming a different type of offspring. So you will be completing a transformation lab. You'll be taking a bacteria and inserting a gene from a jellyfish so that the bacteria, it's a fluorescent gene, so that the bacteria glow when exposed to UV light. It's pretty cool. Most groups do really well, and it works for most. And so hopefully we'll see that. But that's what we're going to be working on in the spring. So what's kind of the basics behind that? Read 1 and 2 for us there, Walker.
Behind transformation.
Mm-hmm.
Okay. So bacteria took in DNA from an external source. Recombination of DNA occurred. Variation created. Biotechnology, this is what we do in laboratories to make bacteria learn new tricks.
So, for instance, we're going to do the one where we create bioluminescent bacteria by inserting a gene. Now, we can insert genes into their DNA. The easiest way to do this is to insert new what? Plasmids. Good. So, for instance, Katie, let's say you are diabetic and your body rejected any cow or pig insulin that you, maybe the normal source that you would get for insulin. Your body's like, ew, I don't want that. And so they're like, well, we're going to have to make some human insulin and we don't want to have to, poke a hole in anybody. So we're going to take some bacteria. We're going to insert a plasmid with a gene, a human gene for making insulin, insert it into the bacteria, provide them with nutrients, and then they will start expressing that gene. And in that way, you can get actual human insulin produced. So that's what he's talking about, genetic tricks and biotechnology. So actually using the bacteria to produce stuff for us. Okay? Transduction. Read down for us, Fife.
This is when new DNA has been carried in by a virus, thus creating a change. A phage virus introduces new DNA into the bacterial. The two DNAs combine into one genome.
So the cool thing about bacterial DNA, usually bacteria don't have a lot of DNA on that one chromosome, but almost all of that DNA, Matthew, codes for something, like some type of gene. Whereas if you look at our genome, which is pretty lengthy, it's estimated that only about maybe 2 to 4% of our genome actually codes for stuff. There's a ton of like old pseudogenes that no longer code for stuff. And there's also a lot of what they think is old viral DNA that's been incorporated into our DNA. This process happens, nowadays where like let's say you get a cold sore, which is a type of herpes virus. So what happens is that virus injects its DNA into your cell and incorporates, it's a retrovirus, incorporates itself from RNA into your DNA. So it's actually making itself a part of your DNA structure. And then when it wants to, when it's agitated, then it expresses that DNA. Okay? And so that's what it's talking about. Like, and you see on page 573, the previous page, you have a bacteria, viruses, they're at war with one another. And bacteriophages are viruses that inject and take over those bacteria and hijack them and basically use their processes to make more viruses. Now, bacteria have evolved a mechanism in order to basically chop up those viruses, incorporate a piece of that viral DNA into their genome so that the next time they encounter that virus, they'll know, hey, this is what it looks like, this is where we need to chop it up and how to break it up. That sounds a lot like what?
Immune system.
Immunity, yeah, the immune system. So bacteria essentially have their own type of inherited immune system. Now, because they incorporated that part of that viral structure into their DNA, what's that going to do for their offspring?
It's going to have the traits.
It's going to have that same viral piece of DNA that it can recognize and fight off and cut up. The recognition of that mechanism only happened recently, and scientists figured out, well, if bacteria can cut and splice in different sections of DNA, we can hijack their system and use that to cut and splice in different DNA into our bodies. And that system is called, it only happened, maybe, I think they figured this out 4 or 5 years ago. CRISPR. Y'all heard of the CRISPR system?
Oh, yeah.
CRISPR is a way of gene editing that we basically just hijack from bacteria. A couple scientists figured out how it works, and we'll get more into that in the genetics unit. So, and the next thing, conjugation. This is the only thing that I've ever been called into the principal's office over. And since I'm posting this on YouTube, I'll be careful what I say here. Although I would appreciate it if you looked up these terms before you turn me in. All right, conjugation. This is what we call bacterial sex. This is the top of page 574. Read number 1 for us, Katie.
Bacteria exchange plasmids, small circular pieces of DNA, through a conjugation tube from the male to female. Bacteria do not have the sexes like humans do.
Who knew? So they exchange DNA through a conjugation tube. Read number 2 for us, Sarah.
F factor, if a bacteria possesses this gene, they are considered male, shown as F plus. F minus are female. They do not possess the F factor gene. Is that what we continue?
Yes. your book says pilus and it says pili. I'm assuming one of these is plural and one is singular, but I don't know which one. Keep going.
This structure is a sex whip for pulling the female close so that a conjugation tube can be made and between the two bacteria. The pili is created by expressing the F factor gene.
All right, so this term F factor, this gene, it's short for saying fertility factor. And if you have it, basically you're male, and if you don't, you're female, if you're bacteria, okay? And this is kind of a loose interpretation of male and female here. But essentially what this structure is, this pilus or pilus or however you want to say it, it is a sex whip. Now, essentially what it's made of, what it is a flagella. You know how flagella, like a sperm, is used to propel you along? If you look up the Latin origin of the term flagellate, it's going to refer to it as a whip. And so basically that's where this term sex whip that the author uses in the notes comes from, because it's acting like a whip from one bacteria to whip out to another bacteria, grab a hold of it, and then through this conjugation tube, you have an exchange of plasmids. Why would you go about doing that if you're a bacteria? Because that sounds like it requires a lot of energy and time and effort.
So you can safely transport the plasma.
And what was the purpose of that, though?
In a harsh environment.
What'd you say, Mason?
It's like if you're in a harsh environment.
So if you exchange those plasmids and that plasmid has genetic information that helps you survive, yes, it could help you survive. It could help your offspring survive in a harsh environment. Good. And that is the reason for this reproduction. For example, if you're a bacteria living in someone and you're able to, you're getting bombarded with antibiotics and you're exchanging some DNA, and maybe you find a way to become antibiotic resistant. Like we hear a lot about antibiotic resistant bacteria nowadays. So those are all bacteria that have evolved that from some type of, gene that's come about, that's been exchanged and that's passed on. Okay? Any questions there? It's all about DNA and survival, and then passing on your DNA to the next generation. The last part, the produce part here, I'm going to go pretty quick on this one because turn to page 600. There's not a whole lot. Hold on one second. There's a good section at the bottom of 575. It talks about antibiotic resistance and when they first realized this, and they talk about R plasmids, R being reference for resistant plasmids. So they're carrying a resistant gene. And so they get a little bit more into that. We don't have time for that. But if y'all want to talk about that or look at that later, you can. So on page 600, there's a couple other organisms here we'll look at. Protists. In your 9th grade book, they probably characterized protists as a kingdom. Did they do that? Did y'all talk about protist kingdom? Okay. This Campbell College biology book does not do that. It talks about protists in terms of a group of clades, which means just a bunch of groups. Protists are kind of a bunch of things that don't fit into other categories, and they kind of threw them in all into one category that doesn't really make a lot of sense. The things they have in common. So you have animal-like, fungus-like, and plant-like protists. Protists are usually small, single-celled. Some of them are larger, though, like some of the bigger algae. They usually live in moist environments, and they are eukaryotes. And that's about the only ways that they go together. You might be familiar with there's a protist that causes malaria. I know you hear about malaria is caused by mosquitoes. Mosquitoes are just the transmission vector, and they actually talk about them on page 599. The thing that actually causes the destruction of your blood cells in malaria is the plasmodium, which is a type of protist. Algae are plant-like protists. They don't have enough characteristics to be grouped with plants. The Irish potato famine was caused by a type of fungus-like protus, which is similar. It didn't have enough characteristics to be classified with fungi. But anyway, so there are a lot of different types of protists. There's a lot of different life cycles. I don't think it's necessary you really get into them. Most of them, their sexual reproduction is similar to ours, where you have sperm and egg that come together to form offspring. Sometimes you have one that's more like fungi, where you have haploid cells. So like fungi, all of their cells that would be in the normal fungi, those are already haploid, but they're not sperm and egg. And so essentially what happens is two of those cells fuse together, and then they undergo meiosis to form sperm and egg. And then they form some spores, and that's where the new fungi comes from. So it's just a little bit different process. So I'm not going to get into all that. I don't think it's super necessary for what you need to know for the AP exam. All right. We are going to hopefully finish up the miracle of life. And you people out there, have fun.